Hi, my name is Aloho, and I'm a graduate of MIT. This video is on proteins. Nearly every dynamic function of a living being depends on proteins. Proteins account for more than 50% of the dry mass of most cells, and they're instrumental in almost everything organisms do. Some proteins help speed up chemical reactions as enzymes. Life would actually not be possible without enzymes, most of which are proteins. Enzymatic proteins regulate metabolism by acting as catalysts. Catalysts are chemical agents that selectively speed up chemical reactions without being consumed by the reaction. Other proteins play a role in defense, storage, transport, cellular communication, movement, or even structural support. A human has tens of thousands of different proteins, each with a specific structure and function. Proteins, in fact, are the most structurally sophisticated molecules known. Consistent with their diverse functions, they vary intensively in structure as depicted by A, B, and C, which are three very different proteins. Each type of protein has a unique three-dimensional shape as well. An amino acid is an organic molecule with both an amino group and a carboxylic group. At the center of the amino acid is an asymmetric carbon atom called the alpha carbon. Its four different partners are an amino group on the left, a carboxylic group on the right, a hydrogen atom at the bottom, and a variable group symbolized by R at the top. The R group, also called the side chain, differs from each amino acid. Diverse as proteins are, they're all constructed from the same set of 20 amino acids linked in unbranched polymers. The 20 amino acids are depicted in this diagram on your right. The bond between amino acids is called a peptide bond. So a polymer of amino acid is called a polypeptide. The physical and chemical properties of the side chain determine the unique characteristics of a particular amino acid, so it affects its functional role in a polypeptide. A protein is a biologically functional molecule made up of one or more polypeptides, each folded and coiled into a specific three-dimensional structure, which we'll talk about later on in this video. When two amino acids are positioned so that the carboxylic group of one is adjacent to the amino group of the other, as it is in this first picture here, they can become joined by a dehydration reaction with the removal of a water molecule, resulting in this right here. The resulting covalent bond is called a peptide bond. Repeated over and over again, this process yields a polypeptide, which is a polymer of many amino acids linked by a peptide bond. Polypeptides range in length from a few amino acids to 1,000 or more. Each specific polypeptide has a unique linear sequence of amino acids. Notice that no matter how long the polypeptide chain is, it will always have one free amino group, and on the opposite end, one free carboxylic group. Thus, a polypeptide of any length has a single amino acid called the N-terminus and a single carboxylic end called the C-terminus. Additionally, the polypeptide backbone is a region in the box that excludes the R chains. The specific activities of proteins results from their intricate three-dimensional structure, which is determined by the sequence of their amino acids. There are 4 levels of protein structure that you should know well: primary, secondary, tertiary, and quaternary. The primary structure of a protein is simply its sequence of amino acids. Take human insulin, for example. Part of its primary structure is glycine, phenylalanine, phenylalanine again, tyrosine, threonine, proline, so on and so forth. That's its primary structure. Most proteins have segments of their polypeptide chains repeatedly coiled or folded in patterns that contribute to the protein's overall shape. These coils and folds, collectively referred to as their secondary structure, are the result of hydrogen bonds between the repeating constituents of the polypeptide backbone, not the amino acid side chains. As you'll see here, the interactions are between the backbone, not the side chains. One such secondary structure is the alpha helix. It's a delicate coil held together by hydrogen bonding between every 4th amino acid, as shown here. The other main secondary structure is the beta-pleated sheet, as shown. In this structure, 2 or more segments of the polypeptide chain lying side by side, referred to as beta strands, are connected by hydrogen bonds between the parts of the 2 parallel segments of the polypeptide backbone. Superimposed on the patterns of secondary structure is the protein's tertiary structure, shown here in a ribbon model of a polypeptide. While secondary structure involves interactions between the backbone's constituents, tertiary structure is the overall shape of the polypeptide resulting from interactions between the side chains, or the R groups, of the various amino acids. One type of interaction that contributes to tertiary structure is hydrophobic interactions. As a polypeptide folds into its functional shape, amino acids with hydrophobic or nonpolar side chains usually end up in clusters at the core of the protein, out of contact with water. Thus, hydrophobic interaction is actually caused by the exclusion of nonpolar substances by water molecules. Once nonpolar amino acid side chains are close together, as these are here in this diagram, van der Waals interactions help hold them together. Meanwhile, hydrogen bonds here between polar side chains and ionic bonds over here between positive and negatively charged side chains also help stabilize tertiary structure. These are all weak interactions in aqueous cellular environment, but their cumulative effect helps give the protein a unique shape. Also, covalent bonds called disulfide bridges may further reinforce the shape of the protein. Disulfide bridges or linkages form where 2 cysteine monomers, which have the sulfhydride group SH on their side chains, are brought together, close together by the folding of the protein, forming this linkage. Finally, quaternary structure is the overall protein structure that results from the aggregation of these polypeptide subunits. As shown in this image, there are 2 subunits of the yellow polypeptide and 1 subunit of the blue polypeptide, which, when brought together, form the quaternary structure of one protein. Not all proteins have quaternary structure, and that's an important thing to keep in mind.